Yeah, so first I'm just gonna give you a little bit of introduction. So my name is Kelsey. I grew up in Texas, near the Dallas area, did my undergrad at the University of Texas in Austin. And I knew I was always interested in biology and science. And I actually got to do my first research experience as a high school student. So I worked in a lab in Dallas studying colon cancer, and I was introduced to cell culture and mouse models and cancer research. And I knew that I wanted to go on to do cancer research based on that. So I went to college, I got my bachelor's in science, and then I headed off to the NIH, where I worked for a year in a super resolution microscopy lab before heading to New York City, which is where I did my PhD at Memorial Sloan Kettering Cancer Center. And that's where I became involved with Rock EDU and their science outreach, and why, even though I'm currently in Michigan, I am connected to you guys in New York. And so in my current lab, I'm, like I said, I'm a postdoc, and this is actually my second postdoc. My first postdoc, I was studying a type of pediatric bone cancer called Ewing sarcoma, and now I'm studying studying colon cancer. And I'll get more into what I'm actually doing research-wise, but that's kind of my career trajectory, and all the different places I've gotten to do research over the years and how I've grown my skills. And so just to get started, I want to ask you guys a very, very important question, which is the first poll. So I'm curious which you guys consider it to be better, pie or cake? Okay, it looks like we have 16 votes in. I think we're waiting for a few more. I'm glad you guys find this as important a question as I do. So let's say another 20 seconds just for a few more people if anybody else wants to answer, or if they want to provide reasoning in the chat, that's cool too. Also, if anybody has any questions already or something they want to ask, feel free. Okay.
Nah, I just want to say one thing. If you say pie is better than cake, you're wilding. You're out of pocket. I'm sorry.
So it looks like cake won. So 70% to 30%, which I find personally surprising because I'm in the pie category. But it's interesting to see everybody's reasoning and where everyone has fallen. Yeah, cake won. Okay. So
Can I give a reason why I say cake is better?
sure, we'll have one.
All right,
cake
so look, let me explain. So with pie, it's like you have such limited things, but with cake, you got stuff like cheesecake, you got red velvet, which is the god of all cakes. So you have so many different options compared to how pie is like you got apple pie. I won't get you wrong. You do got sweet potato pie. You do got pumpkin pie, but like with cake, you just more stacked and overpowered over there. In my honest opinion.
That's some very interesting reasons. I guess I have to sample some more pies in order to fully have a good scientific experiment with a large sample size.
I really appreciate that.
So yeah, so now I'm going to switch over to my phone. So just give me a second. And we're going to go on a lab tour. Okay. So can you guys hear me now?
Yes, we can.
Yeah.
Okay, cool. So I'm currently, like I said, I'm currently in the cell culture room just because with COVID, we have some space restrictions. And so this way I'm out of the way, but I'm going to take you guys out into the rest of the lab. So you can see here is like some benches. So this is where we work. So people are doing various experiments. So you can see more bench spaces. We have lots of fridges because we have to store things at various temperatures. This is a room where we do a lot of experiments that involve running gels. So if we need to like look at protein levels or RNA levels or DNA levels, this is the room where we do a lot of that work. We also have our is where we work with mice. And they're a good model for cancer because they're similar to humans, but you can obviously do things with mice that you can't do with human subjects. Yeah, so this is kind of our lab space. We have another cell culture room over here. You can see we have someone currently working in the hood. So she's splitting some cells. You can see how it works. This is Christina.
Hi, Kelsey. How are you?
Good. Yeah, so basically the basic components of a cell culture room are the incubator, the hood where you work to keep things sterile, and your microscope. So we're gonna head back to the other cell culture room where I'm gonna actually get some cells that I'm gonna be able to show you guys. So yeah, as I said, this is the hood, and basically it works by you raising up the sash, and then you can work inside of it, and inside is sterile, outside not sterile. So this is how we prevent our cells from getting bacteria or fungal contamination. We grow our cells in an incubator. And so this is an incubator. We keep it at 37 degrees Celsius, which is about 98.7 degrees Fahrenheit or body temperature. So basically these cells are used to growing in a human body, so we keep them at the same temperature. So you can see we have a whole lot of different plates of cells all throughout here. So I'm going to get some of my cells out, and we're going to go next door to the microscope. So normally I would look at it on this microscope, but it's a little hard to show via the phone. So we're going to go next door where there's a camera connected to the microphone. Sorry, a microscope, not microphone. So this is the microscope, and it has a camera connected to it. So on the screen, once I turn the light on, we'll be able to see cells. And so I have a few different types of cells for you guys to look at. So this first one is a colon cancer cell line. So you can see that this is what the cells look like. They're growing in a dish. You have this red liquid on top, which is just their media. Basically has all the nutrients they need, but the cells are really small. So in order to see them, we need the microscope. So we're gonna turn the light on, and then we have to focus on them. So you can see these are our cells. So basically all of these little shapes. They're kind of like a weird triangle or like an elongated like diamond and so we have all the different cells that are growing, and then we have the cell like places where there are no cells. And so this is a colon cancer cell line. So it grows attached to the plate. So it's adherent. So you can see as I move the plate, the cells move. And there's a few cells that are kind of floating. Those are dead cells. On the other hand, I also have this other cell line right here, which is a suspension cell line. So it grows in suspension. It doesn't adhere, it doesn't attach to the bottom of the dish. And so you can see when I move that basically the cells move back and forth. And also that, you know, there's a few different levels at which I can focus. And so these are a cell line that were made from red blood cells, basically the precursors to red blood cells. And so what they did was they added in a gene that allows these cells to grow forever. Because normally your cells only grow for a certain amount of time and then they stop. And by adding in this gene, we can grow these cells forever. So we can use them in the lab in order to study red blood cell differentiation. We can see how if you take something away or you add something different, how that affects differentiation. But you can see that these cells, like I said, are suspension, whereas the other ones were more attached. So for both of these, I showed you the image in bright field. So this is basically just normal transmitted light. Something else that we can do to our cells is we can add in fluorescent tags so that then our cells have color to them when you shine the right laser light at them. So that's what these are. So I'm going to turn the light off real quick and switch over to the GFP channel. So you can see here that you basically have these cells that have green in the circle like this. So these are cells that have GFP or green fluorescent protein attached to their mitochondria. So basically all of their mitochondria, which produce the energy in the cells, are labeled with GFP. And so you can use these to study the mitochondria. For example, if you treat the cells with a chemical and it causes an increase or decrease in the green color, or that changes the patterning of them. So yeah, so that's how fluorescence can be used in microscopy and cell culture. So we're going to head back to the main lab. But while I'm getting resituated, if you guys can start poll number 2.
On it.
Thank you.
All right, so I'm about to launch the next poll question. So the question is, the fact that some cell lines grow in suspension while others attach to the plate is based on, and there are 4 different options. I'll give you guys a minute or 2 to answer.
And so I didn't specifically go over the answer, but think about what I showed you, what type I showed you as suspension and what type I showed you as adherent, and see if you can reason your way.
And it's okay if you don't know, just choose like the best answer you think it is.
Yeah, I honestly don't expect you guys to know the correct answer. This is just to see your reasoning and see what your guess is, and then we'll talk about it.
We have about 50% voted. We're waiting on the other half.
So I do want everyone to participate. Again, it's based on your reasoning. Please participate.
And I don't know who answered what, so there's like no grade associated with this whatsoever.
Yeah. And I'm looking through the chat. while you guys are answering. Yeah, so the question about forever growing cells. So that's kind of how you get cancer. And so like, but some of these same genes that you can put into your cells cause you to get cancer, which is basically your cells growing forever. So a human living forever would not really be as possible. But the cells themselves, yeah, that's it. That happens normally inside your body if you get cancer.
Yeah, so it's like a tumor. That's why you end up getting tumors, because the cells are just continuously growing. Awesome. So I give you 10 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1.
Okay, so can you guys hear me? I turned off my phone and okay. So yeah, so I'm gonna share the results. It looks like you guys were split between whether they come from cancer, normal tissue, or how they normally grow in the body, which actually are really good guesses. Because basically I showed you a colon cancer cell line that grew adherently, and I showed you a red blood cell line that grew in suspension. And so I could see how, you know, normal or cancer would be a good guess, but the correct answer would be how they normally grow in the human body. So if I had a blood cancer cell line, for example, that would also grow in suspension. So blood, you know, kind of, it, you know, it's suspended, it's liquid inside of you, and the cells are kind of just floating around, as opposed to your colon, you know, your large intestine, where the cells are growing, you know, attached, like, you know, they're a sheet. So the same thing if you had like a skin cancer cell line, that would grow adherently. And so most cell lines grow adherently. It's more the blood ones that tend to grow in suspension, but they you just have to figure out the best way to culture. Every cell line's a little bit different. So yeah, so does anybody have any questions about anything I've shown you with cell culture so far, or anything at all before I move on? Okay.
Hold
So
on,
now I'm going to tell you guys.
I'm sorry.
Huh?
I just want to reinforce that, you know, like, you should definitely ask questions. This is going to be as interactive as you make it. So again, definitely ask your wondering questions that you have.
Yeah, you have a scientist at your hands right now, so take advantage.
Yeah, and there are no stupid questions. Like, please ask me, you know, anything, I guess science related, but, you know, like, I love help. So it looks like we have Mary Ellis. Is that how you pronounce your name?
Yeah.
So what's your question?
So isn't cancer like when the cells like have a miscommunication and there's an error that happened?
Yeah, so it's often caused by mutations in their DNA. So when, you know, you have your DNA, it's being replicated, and, you know, you have your base pairs. Instead of like putting an A, for example, you end up with a G or something. And that causes the gene to now either lose its function or gain extra functions. And so this happens a few different places in the DNA. And together, they allow the cells to proliferate, to grow uncontrollably. So they basically, normally in your body, there's a lot of regulation of, like, you know, cells can grow and then they have to stop. For example, like, say you have a wound, you need to regrow a bunch of skin cells in order to fill in the wound. But once you fill in the wound, the skin cells stop growing. But with cancer, there is no stop signal. They basically have stopped listening to the rest of the body. So they just keep growing, keep growing, keep growing. And that's how you get the big tumorous mass. Did I answer your question?
Yeah, thank you.
Yeah. So let's see. I think we have a few in the chat. So we have somebody who asked about is stem cell research still relevant in the labs? Yes, stem cell research is extremely relevant. You know, a lot. So I'm mostly focused on cancer, but I also do some research looking at development. So how red blood cells differentiate. And so for me, I use not quite a full stem cell, but like a little bit more along the way, and then I induce it to differentiate into the red blood cell. But a lot of what we're understanding about how the body forms from basically from fertilization is using stem cells. We're also figuring out how to potentially treat diseases. So stem cells are still very much a relevant field. Let's see. Have there been experiments trying to take the forever expanding properties of tumors and repurposing them? Andy, would you mind telling me exactly what you were trying to ask here?
Well, like you see how tumors can expand forever, right? So that would mean, well, I'm assuming that would mean that this, like the tumor mass itself can live forever, I guess. Right. So would they take
Yes.
like some of those properties of the tumor itself and like for example, that it can expand forever or other properties that it may have and using them for other things or like, I don't know, trying to use them for like medicine or something like that?
I mean, I think definitely everything we learn about how cancer works helps us understand how things go normally. Because, you know, if something is happening abnormally, you can suddenly discover certain signaling pathways or interactions between different cells that are relevant, you know, under normal developmental or normal cases, and you can figure out, you know, how they are relevant. Same with, you know, certain other diseases share, you know, properties with cancer. And so if you can understand how this one pathway is misregulated in cancer, you can understand, you know, how it's misregulated in this other disease. And but as far as like taking their ability to replicate, at least I'm not aware of how that's been used other than in the lab where we have certain cell lines. So like, you know, if I took a sample of my blood and I tried to grow it in the lab, it would die pretty quickly because the cells can't grow forever, and also I'd have to have the right nutrients. If I took the genes that cancer, you know, uses to grow forever and I put it into my blood cells and tried to grow it in the lab, then it would work a lot better. And so it's more using it as a tool in order to make these cell lines that normally would have a limit, you're able to grow them forever. It's called immortalization or transformation. But I don't know about any cases where it's been used beyond that. But that doesn't mean that it's not. It's just I'm not aware of it. It looks like we have one final question. So is chemotherapy supposed to stop the cell growth? Yeah. So one of the hallmarks of cancer is that it grows a lot faster than all the other cells in your body. And so chemotherapies basically act upon that. They kill cells that are rapidly proliferating more than they kill cells that are not growing as fast. And so the cancer is more hit, but it's the reason why if you treat with chemotherapy, you lose your hair, you can have gastrointestinal issues. And that's because the cells that are producing your like hair follicles or the cells in your gut also rapidly proliferate. And so you're hitting cancer, but then you're also hitting these other cells, and then the rest of the cells in your body are kind of spared. But that's why you have these side effects. It's very much a blunt instrument for treating cancer, although it's, you know, done wonders and was initially how we started treating cancer. Now we're starting to be a little bit more targeted by using things that are specific for the mutations in that type of cancer, or immunotherapy, where you take the person's immune system and basically target the immune system towards the cancer. And yes, the hair does grow back because once you remove the chemotherapy, the cells are able to come back because you have the stem cells. And potentially the cancer can come back depending on if you got rid of all of it or not. And so
Yeah, I was about to ask you, like, so if like one cancer cell remains, it does come back, right?
Yeah, so I mean, if you can't get rid of all of the cancer cells, then it can come back. And so that's part of the reason why we try so many different ways of treating. So sometimes somebody will get surgery and radiation and chemotherapy because, you know, the surgery removes the bulk of the tumor, then the chemotherapy and the radiation can help get those cells that are remaining. And if they get all the cells that are remaining, you're good. If they don't, which it's sometimes hard to tell if they do or not, then that's when you have relapse down the line. And so, you know, one of the major focuses of cancer research is trying to get better and better at getting all of those cells, that, you know, targeting every last cell before you stop the treatment. Although, you know, the treatment's limited. You can't treat somebody with chemotherapy for the rest of their life because, you know, the toxic side effects, and it possibly could have other effects down the line. Looks like Rosalind has a question.
Yes, so I wanted to ask about breast cancer because I have seen multiple, articles and also news that talk about that people of color, well, women of color, especially Black women and Hispanic women, are mostly affected by breast cancer. And majority of the time, Black women are end up actually having develop more aggressive cancer and also stage, and which is why they're diagnosed at a very young age and also end up die very quickly compared to white women. So I would like to know why, maybe.
So I'm not an expert at breast cancer research. From my understanding, I don't think we fully know. I think some of it has to do with other genes that may be expressed, in Caucasian versus Black women. But yeah, I mean, it's definitely a major issue that cancer is not one disease. Cancer, basically every single person who has cancer has their own unique disease. Like the mutations are different, the overall person is different because, I have certain genes that my body, or I have certain, alleles of genes that someone else doesn't. And we don't entirely know how that plays with the mutations in the cancer. And so I'm sorry, I don't have a better answer other than, yes, that is something we are very much aware of, that there are these differences, but we haven't yet fully figured out what causes them. Mary Alice?
Okay, because the mutation, because cancer is a mutation to the cells, is that why it's able to be passed on? Like that's why, like, if one of your family members has like a certain type of cancer, it's a higher chance that you're gonna get it.
So sort of. So there are different contributors to cancer. So there's, environmental effects, for example, smoking. There are, genetic effects, and then there's just kind of random chance. And so if I have a mutation, for example, skin cancer arises from moles. So I have, a mutation that occurs in this mole on my arm that causes me to get melanoma. I'm not going to pass that on to my children because that mutation arose spontaneously in this one skin cell. However, if throughout my entire body I have a mutation, for example, in BRCA1, which is associated with breast cancer, and that's throughout my entire body, which includes my germ cells, so my eggs, when I have a child, there's a possibility of me passing that on to my child, and depending on, the child's father, what their genetics are, then I can pass on an increased risk of cancer to my child. And so it basically depends on where the mutation is. If the mutation is somatic, so it's only in normal body cells, so not your egg or sperm cells, it won't be passed on. If it's throughout the entire body or it affects your eggs and your sperm, then you can inherit it. Yes, and those are good points. And then why do autoimmune diseases attack the body's healthy cells, and how does it get treated? So I'm not an expert on the immune system, but from my understanding, so you normally have your T cells, and they're trained to recognize self versus non-self. And the ones that recognize self, the ones that recognize like me, are weeded out, they're killed. And it's only the ones that don't recognize self that that don't recognize self that are able to continue growing inside of your body, and they recognize bacteria, viruses, cancer cells. But in some people, the self-recognizing cells, for whatever reason, are not killed in that same way, and so then they are able to attack your body because they think that it's a foreign invader, basically, because T cells don't know self or self, they just know whatever they're designed to target. And so if whatever they're designed to target happens to be you, they're going to attack you the same way that they would attack any other pathogen, any other foreign invader. So any other questions? These have all been really great. I'm glad you guys started asking questions. Okay, so I'm going to start talking about what I currently do. What I have about 5 minutes left.
I think give or take.
Okay. That's fine. I mean, like I said, I'd much rather we have this discussion. So I guess before I do it, if anybody has another question, I would love to answer it. If not, I'll just give a very brief overview of the two things that I study.
Is there a specific time that y'all have to leave?
Yes, their next class starts at I think it's 12:28.
Okay, 12, 5 to 6 minutes. Okay, cool.
Oh, and before I forget, I gave your teacher my email address, and it goes in the bio. Please feel free to contact me if you have questions about like how to get into doing research. If you have science questions, I may not always know the answer, but if I do, I would love to help. So please email me with those questions if you have them. So as I said before, I study colon cancer. So it's, your large intestine, cancers that arise there. And kind of two major aspects of it. The first is trying to study the role of red blood cells in cancer. And so, if you consider the cancer itself, as this small community of cells that are all cancer cells, there's also all these normal cells that interact with this cancer, that surround it, that are all around it. And one of the cells includes red blood cells. And so we're interested to see how red blood cells can interact with the cancer cells and promote the growth of the cancer. So we think that they might provide nutrients and maybe other signals that promote the growth. Go ahead. I mean, I have like 5 more minutes. Okay, sorry. So that's kind of the first thing that I'm trying to study, is trying to see how the normal cells outside of the cancer interact, because we're becoming more and more aware the cancer microenvironment, this environment of normal cells, actually plays a very important role in cancer. It's not just the cancer cells in isolation. And potentially we can, instead of just trying to target the cancer cells, we can treat and try and target these normal cells, prevent these normal cells from giving the cancers whatever nutrients they need. And that's another way we can possibly try to treat and cure a person's cancer. And then the other project that I'm focusing on is looking at the role of glucose, which is a sugar, in colon cancer. We know that cancer cells actually metabolize glucose much faster, or much more rapidly compared to normal cells. And this has to do with how they're metabolically set up. And so we're trying to see how in colon cancer, glucose affects the signaling pathway called the STAT pathway, STAT3 pathway, and how that promotes the growth of the tumor. And so if we can understand how glucose leads to activation of this pathway, which promotes the growth of the cancer, then maybe we can once again come up with new and better ways to target colon cancer. Because colon cancer is one of these ones where we can screen for it and we can treat it early, which is good, and that's part of the reason why it's highly curable. But then there's other people that whatever we've tried to this point, it just doesn't work. And so we're always looking for a new understanding of cancer so that we can have a new therapy to try. And so I'm not involved in the creation of the therapies, but I'm trying to have a better understanding so that way other people can take that understanding and convert it into therapies. So yeah, that was a very quick overview of kind of what I do in the lab. So I guess before you guys have to go, any last questions, comments? I'd love to talk about anything else that people are interested in. Okay, well, I mean, do I enjoy working in the lab? Yeah, I very much enjoy how doing research combines being hands-on, like I get to work with my hands, but I also am working with my brain. You know, I'm planning experiments, I'm thinking through results, but it's not just sitting in front of a computer all day. I enjoy, actually doing things, even if it's just transferring liquid from one container to another container, which, it's actually doing something, but, at the time I can't see, but then I get this really cool result and then I get to interpret it. And so, obviously COVID has, affected working in the lab. Now we all wear masks and we have to keep our distance and only a certain number of people in the room. But yeah, overall, I think research is a really cool career. And I'm really glad that I get to do it. And I'm really glad that I've had the opportunities I have had that led me on this path. Yeah, so if you want to get started in a science field, so like I said, I was fortunate as a high school student to get to work in a lab one summer, and there are some programs where high school students can join a lab for the summer and they learn. And I guess I can mostly speak from biology. I can't speak of other sciences because that's not what my experience is in. But I think, either as a high school student or as an undergrad, a lot of people do it as an undergrad, getting into a lab either via the summer or during the semester. There's a whole bunch of different programs at various research institutions at universities, applying for those. Or once you go to college, like you're in biology 101, you can go talk to your professor and be like, hey, I want to do research. Like, do you have any suggestions on who I should talk to, or, what lab I might be able to join? Also, as was pointed out in the chat, Rock EDU has a lot of great programs that introduce students to research. But I think a lot of it is either trying to find a program or just sending an email asking someone. That was how I got my first research experience in a lab. Was I emailed this one professor and I was like, hey, can I come work for you? And he didn't have room, but he passed my email on to somebody else and they did have room. And so I ended up in a lab that way. And it was such a really cool, great experience. I think the earlier you can start, the better, but it's also never too late to start. So, I think it's all about exploring. I've done research in a bunch of different topics. I did plant research for a year while I was an undergrad, and I quickly learned that I didn't want to study plants. And so I think just trying things, figuring out what field you're interested in. For me, it's cancer biology. I like trying to understand how these cells take what happens during normal development and manipulate it and turn it to their own uses. But for other people, it could be immunology, it could be material science, it could be physics. Just finding what research, if that's the career you want. There's also so many other careers. So just try things, I think, would be my biggest advice.